Well, welcome everyone. It looks like we've got a large crowd today. It's wonderful to see this size group. So thank you for coming. My name is Wayne Stacy, and I am the Executive Director for the Berkeley Center for Law and Technology. For 25 years, BCLT has been the focal point at Berkeley Law for issues at the intersection of law and technology. For 15 of those 25 years, Berkeley has been rated the top intellectual property program in the country. BCLT's scope, however, as you're going to see today, covers more than just traditional IP. For example, we cover privacy, computer crime, cybersecurity, biotech, all life sciences, and technology regulation. When you think of BCLT and Berkeley Law, your mind naturally goes to student education, but that's just part of our mission. We also partner with leading law firms and lawyers to explore cutting-edge legal issues and to share that work with practicing lawyers. Today's program is a perfect example of that public-private partnership. Morrison Foerster is a firm that we have worked with for many, many years. Today, they are leading a program on legal considerations in commercializing gene-edited plants. Mike Ward will be moderating and driving this discussion for us today. Mike is the head of MoFo's patent and life sciences practice. He is also the co-chair of the firm's food and ag tech practice. If you look at Mike's group, it spans the business activities all the way from M&A to private equity to emerging company work. And not to embarrass Mike, but when you look at his list of accomplishments, he's among the most awarded and recognized attorneys in the country in the life sciences field. So on behalf of BCLT, I want to thank Mike for accepting our offer to organize and plan today's seminar. These types of events take a lot of time to put together, and Mike and MoFo as a whole has been very generous with their time. So thank you, Mike, thank you, MoFo, and thank you all for coming today. Mike, I'll turn it over to you.
Great, thanks very much, Wayne, for that kind introduction. I think we have a very interesting seminar planned for you today and tomorrow. And as we began to put our presentations together, we soon realized that this was a pretty ambitious agenda. Many of the topics that we will cover over the next few days could be the topic of a seminar all on their own. And so for those interested in deeper dives into one or more of these topics, you'll be pleased to hear that we have additional seminars already in preparation. But that being said, we expect the next 2 days to be packed with useful information. If you have questions during the presentation, please use the chat function. And we will get to those today and tomorrow. What our plan is today we'll go for an hour as we promised. And then tomorrow, in order to get to the questions that we anticipate from the audience, we will go an extra half hour to answer any questions. So we'll end at the hour mark. You know, folks can obviously drop off whenever they want, but if you want to hang around for the second 30 minutes after tomorrow's session, we're going to be doing Q&A. So today's session, we are very lucky to have Steve Delaporta from Yale University and Veranomics, who's going to talk about the technology and then give us a hypothetical which will guide the discussion for the next 2 days. Then we'll have Otis Littlefield from MoFo, who's going to talk about freedom to operate issues. And then also Matt and Wolfgang and Otis will talk about licensing issues today. Then Brooke on the next slide. Tomorrow, I will talk about IP strategies. And then Bill Tarantino will talk about regulatory issues for gene-edited plants. Wolfgang Schoenig will cover European regulatory issues for gene-edited plants, and Bethany Hills will talk about regulatory issues for food produced from gene-edited plants. So we have a very packed agenda. Again, use the chat function. With that, I want to turn it over to my friend, Steve Delaporta from Yale and Veranomics. Steve?
Thank you, Mike. And just to check, everyone can see the screen and hear me. Any problems? Good. All right. So first of all, thanks for the invitation to talk. I have sort of a difficult task to spend the next 15 minutes explaining gene editing technology. And I've decided I'm going to really have a sort of realistic an agenda and present a very brief scientific overview of gene editing and its application in agriculture, state the problem a bit, what we're facing, gene editing approaches, but restricting my discussion to the CRISPR-Cas9 system, and then provide a hypothetical or an illustrative scientific technology application. So first of all, in agriculture, we're facing monumental challenges. Basic human nutrition and food security with a growing population. We still have 3 million children dying each year due to malnutrition, another 160 million suffering from some form of malnutrition. We have scarcity of water resources in arable land. And as you know, you've heard in the news that the West is facing historic droughts this year with anticipation of over $200 million in losses due to this to the drought out west. And so, these are issues that we're facing in agriculture. And if you sort of compound that with climate change and the shifting cultivation zones for key crops, you can see that we really have quite a bit of work ahead of us. But the bad news is, there's lots of issues. The good news is there's technology around the corner that's really going to have a major impact, and that's gene editing. One of the things I'd like to sort of illustrate is emerging agricultural pandemics. I think it's a sort of a topical issue. And agriculture faces the same pandemics that other species face. There's a solution to these problems, and that is we increase production of more nutritious food produced with less chemicals, fertilizer, energy inputs, soil erosion, and water. Simple problem, right? Of course, the technology behind that is somewhat complicated. We have conventional breeding, but this is a decades-long process of varietal development, which for some rapidly growing crops can develop new varieties in a matter of years, but there's limited crop diversity. So in our commercial germplasm, no matter what crop it is, there's quite a bit of diversity that's been limited through the process of domestication and breeding. We have GMO technology, and as you know, there are regulatory hurdles, there's great expenses, and there's consumer acceptance issues that we have with GMOs. And finally, we have precision gene editing. And there's a clear path to deregulation. It's a non-GMO technology. And if it's done right, I think consumer acceptance will be there, because a lot of this technology can be applied to making food more nutritious, that is consumer traits rather than just agronomic traits. So this really begins with work on bacterial innate immunity systems. And I don't have time to explain that, but as you probably know, last October, Emmanuel Charpentier and Jennifer Doudna at Berkeley was awarded the Nobel Prize for the development of gene editing methods. And why this is so important is that we're really on the cusp of being able to make any desired genetic modification at any genomic position in a bacterium, plant, or animal cells without unwanted genome modification. I really do believe that we're just a few years away from realizing that ultimate goal. So what are the applications? First, nutritional fortification. We can edit and create more nutritious foods with essential metals and vitamins and antioxidants. So consumer traits such as those. Agronomic traits, plant architecture can be modified. And as you may know, the Green Revolution, one of the major features of the Green Revolution is to create more upright leaves, which allowed planting density to increase. That gave rise to increased yields. Nitrogen usage, biotic and abiotic stress resistance, consumer traits, shelf life, colors, flavors, next generation plant-based proteins. These are all targets that are going to be addressed using gene editing technology. If we look at water efficiency, modifying root structure, stomatal density, and creating drought-tolerant crops is within reach. And finally, carbon mitigation, carbon sequestration, biofuels, and reforestation. Projects such as reforestation, areas that are waste areas or deserts become tractable with this type of technology. So I'm going to focus on emerging pandemics as an illustration of how this might work. So first of all, let's look at the product development pipeline as it exists sort of traditionally. We have new traits that we bring into our commercial varieties using breeding technology, mapping populations, we map traits, we develop markers, phenotype, produce foundation certified seed, and eventually release a commercial variety. That can take 6, 7, or more generations for that process, for the development of a single variety. What's really remarkable, and it's really, I think, just absolutely astounding, is with gene editing, once the trait gene is identified, editing technology can be applied to each commercial variety. And so you're basically accelerating this process tremendously. And I think that's really the ability to create these edits and introduce traits into pre-existing commercial varieties is really part of the excitement behind gene editing technology. So let's talk about pandemics. We've been domesticating for centuries, for thousands of years, wild species and creating food crops. And that traditional breeding effort represents a genetic bottleneck. We've also further bred elite inbreds and hybrids, which further has created a bottleneck, a reduction in genetic diversity. So while we've selected for higher yields, we may have also created greater susceptibility to emerging diseases and environmental stresses. And that's what we see. We see this in bananas, right? If we think about bananas, the Cavendish bananas, there's a blight that's wiped out Cavendish bananas in Asia, Australia. It's been introduced in Latin America, and this may soon be an extinct commercial species. And when you're dealing with especially vegetatively propagated crops, where you have a monoculture of genetically identical plants, this is really an issue. And so we have to approach these problems and think about how are we going to introduce diversity back into these species. The tomato rugose virus is a virus where there's no known resistance in the tomato germplasm, and there's devastating losses that can occur, especially in the greenhouse environments, where the only known solution is just basically destroying the crop itself. So how are we going to come up with resistance to something like this? Wheat rust, this is UG99, which is a wheat rust that was first discovered in Uganda, and it was predicted to wipe out 90% of the wheat crop worldwide, leading to widespread famine. And it was only until outside of the gene pool was resistance discovered to be able to combat this disease. And the one I want to talk to you about is citrus greening. And citrus greening is a disease, it's a bacterial disease, it's a vector-borne disease, it's a psyllid insect that introduces this bacteria into citrus trees. And this is a $5 billion commercial losses in Florida alone. This is a major disease. It's wiping out. It's a devastating disease that's really having a major impact in Florida and is now appearing in California. So if 10 years ago, this is driving through central Florida, you would see these beautiful groves, and you go back a few years later and most of them have been severely affected by citrus greening. So it's gone from 800,000 acres down to less than 5,000 acres, and it's expected to drop even further as this disease runs rampant throughout the Florida citrus industry. Now, what happens when this disease is introduced into the plant, these large, sweet-tasting, fleshy fruits become small, bitter-tasting, dry fruit, and so they have no commercial value. And that's due to the Wang Long Bing, or HLB, bacterium disease, which is also referred to as citrus greening. So when you're dealing with just Florida alone, a $9 billion industry, and there's no known resistance in the citrus gene pool, we're dealing with a real severe problem that needs to be addressed through non-conventional methods. So this is the psyllid that spreads this, and what's interesting about this vector is that it mutates so rapidly that within a single year, it can generate resistance to chemicals. So it's a problem that really doesn't have a solution as we know it. Now, why is that? So if you think about the loss of genetic diversity, citrus was cultivated thousands of years ago in Asia from wild trees that were growing in the forest. And this domestication process that took place selected for desirable traits, which are shown in green, and selected against undesirable traits, which are shown in red. Well, we have our modern citrus, these productive trees that are grown essentially in monocultures. And this can pose a problem because what wasn't selected for was resistant to citrus greening. So even though it may exist in the wild, it wasn't brought into the citrus germplasm. And all commercial citrus is susceptible to this disease. So if you look at sort of the evolution of citrus, the three basic commercial species, grapefruits, oranges, and lemons, are all susceptible to citrus greening. Yet there's resistance when you go to the wild, you see that the genetic diversity exists and resistance exists. So how can we incorporate resistance into commercial citrus? Well, we can't really just cross it in from the wild, because remember, we'll be reversing that domestication process, introducing undesirable traits along with resistance. And so we're going to have very low commercial value, decades of breeding that would be required to generate a commercial crop that was resistant. So what about gene editing? Can we use gene editing as a method to do this? And the answer is we can. And so I won't go through all of the details here, but if we think about gene editing, the first phase is to identify the target, and that itself can be a formidable task. The second phase can be divided into two sort of parallel tracks. One is the design and testing of guide RNA vectors and delivery methods for the CRISPR complex, and also the tissue culture procedures, the cell culture transformation and regeneration that's also required for this type of editing to take place. In the second phase, of phase 2B, is often underestimated, how much work that can be for recalcitrant crops. So we start with resistance trait, a gene is identified, we validate it, we edit it, we create resistance plants from the edited cells, we propagate and commercialize. That's sort of the timeline of creating resistance. So just to quickly go through sort of an example of this, and this is just an illustration, imagine this citrus plant that's susceptible to citrus greening develops a somatic mutation or a sport, which is now resistant. We could take that sport and vegetatively propagate it and develop a single resistant cultivar from that event. Or we can use that sport in a breeding program and decades of conventional breeding to develop multiple cultivars. Or the third possibility is we identify the resistance trait and we edit those directly into commercial varieties, shortcutting that decades-long process. Now, the trait mapping itself can be very complicated, and I don't have time to really go into the details, but you can imagine with current genomics technologies, we have the ability to look for changes and associate those changes with traits such as resistance. So in a case of a sport, you're dealing with a somatic event, you can take the reference genome, you can sequence the variety, you can identify millions of genetic variants that are both common and unique to that variety. And within that pool, there's going to be sport-specific variants that are found in the resistant sport, and that may represent tens of thousands of variants. And through a number of machine learning and statistical methods, you can focus that down to a single gene. So that process may take quite a bit of effort, but you can identify genes. Once the gene is identified, now you can apply editing technology to try to recreate that resistant gene in these commercial varieties that now contain a susceptible allele of that gene. And there's different ways of doing this. And again, this is sort of a field in itself. And I refer you to this excellent review that was recently published out of the Liu lab. But there are the basic nucleases, such as Cas9, which and then various derivatives, such as base editors and prime editors that have been where the basic Cas9 machinery has been modified in order to accomplish other types of edits in the genome. But again, the goal here is to install any genetic change at any position within the genome with minimal unwanted genome modifications or cellular perturbations. So just focusing on the simple case of the Cas9, if we use Cas9 and we develop guide RNAs to target that susceptibility gene in citrus, we can use the Cas9 machinery, that guide RNA, to hone that machinery to the target and the nuclease part of the Cas of the ribonuclear complex to create a double-stranded break in that DNA. That double-stranded break is highly reactive, and there are cellular mechanisms to repair that break that introduce heterogeneity at that double-stranded break. And this is a process called non-homologous end joining. And those bases that are inserted or deleted at that break can result in gene mutations, such as loss of functional alleles. There's another process by which you provide homology to that when you create the double-stranded break, and you can actually knock in a gene, which is called gene replacement. That is a very inefficient process, especially in plants. And so still technology that needs to be improved before that becomes routine. So we have a susceptibility allele, your favorite gene, right, that you want to mutate. You develop the guide RNAs, you test the guide RNAs, you introduce these double-stranded breaks, you allow non-homologous end joining to repair that break, and you can now select for resistant alleles. And in this case, this example I'm showing you, we're just looking for a simple loss of functional allele. So a lot of the repairs that take place would result in frame shift mutations and so forth. Once you're able to do that, think about the fact that you can edit that trait using into a commercial variety and then edit it into multiple commercial varieties simultaneously without having to go through this arduous breeding technology. You can also use that same guide RNA system to edit that trait into the 3 major species of grapefruits, lemons, and oranges. And finally, there's a lot of other editing targets. Citrus gene editing has been enabled, and there's many different targets that could be addressed in terms of sexual or asexual reproduction, fruit quality, fruit color, other diseases, and flowering time. These are just a sampling of the types of traits that are amenable to gene editing in citrus. So the last thing I will say is that there's a minefield here, right? There's a lot of different editing systems, and choice of editing systems can be the difference between success and failure in this experiment. There's different editing processes, non-homologous end joining, the one I showed you, homology-directed repair, which is a gene replacement. There's base editing, there's prime editing, and so forth. There's tissue culture technology, media, hormones, and supplements that have to go into this. There's delivery methods. How do you get that CRISPR complex into the cell for editing through a technology such as agrobacterium or biolistics, DNA-based transfection, RNPs, viral vectors, and so forth. There's the target cells. What cells are you going to edit? Cell culture, embryos, pollen, leaves, these are all possible. And finally, there's the vectors, the Cas9 nucleases, Cas12 nucleases, promoters, terminators, introns, nuclear localization systems, and viruses. And then we mentioned the difficulty with recalcitrant species of regenerating these cells, which may require morphogenic regulators such as baby boom and Wurschel just to regenerate the edited plant from the cells that you create. So again, I try to keep that within my time frame. I hope I wasn't going over too long. I want to thank both Morrison and Foerster and Berkeley for the opportunity to introduce this topic to you today. Thank you.
Thanks, Steve. That was great. We're now going to turn to Otis Littlefield. We can get his slides up to address the freedom to operate issues. All right, you've got this great technology, and you want to use it. You know, what are the sorts of licenses you're going to need? Otis?
Thank you. Okay, so as Stephen alluded to, there are legal minefields, and freedom to operate is the term for what we say is when there are no legal minefields, as in you have freedom to operate because nobody has a patent that is issued in your jurisdiction that can block you from doing whatever it is you want to do. So how do we help you assess if you've got freedom to operate? Well, you come to a patent attorney like myself or my board. We will help you search the patent database for potentially relevant patents that have issued or patent applications that could issue, that could block you. Once we've searched the databases, refined down to a small subset of these are the ones that look problematic, We will then sort of work with you to figure out, is there a way of changing? Is there a way of redesigning your experiment, your product, your screening assay to avoid the issued patent or the patent that might issue? That's one way of getting freedom to operate. Another way of getting freedom to operate is looking at seeing, can you do this in another country where maybe the patent hasn't issued and won't issue? And that could be another way of getting freedom to operate. Another way of getting freedom to operate would be to challenge the issued patent to say, this patent should never have issued, it's too broad, there's prior art, or whatever reason you want to eliminate the patent as a problem. And then finally, you can license the patent, and that is another way to get freedom to operate. And so, again, we can help you with this and figure this out for you. What I often hear is, what does a freedom to operate cost? How much will it cost to get freedom to operate? Well, the issue is that's hard to say. You never get to 100%, you are guaranteed you have freedom to operate. It's really, how much money do you want to spend to get to your level of comfort? And that can be based upon the economic opportunity, the likelihood of you getting sued. There's all kinds of factors that go into. And so if you ask me, how much is this going to cost? I can design a search for CRISPR space that'll be $1 million. That's probably not what you need. I can probably design one that's focused and particular to your system that might be more like $20,000. And again, that's where you need to work with your patent counsel to figure out what's the appropriate search to get you the level of comfort that you have freedom to operate, to meet your goals, to get your investment, to get your product out there launched, to get your interesting new technology developed, whatever your goal is. And the way we tune these searches is we use keywords in searching. And so we can use more keywords, broader keywords. We can search for nuclease, not limited to CRISPR, not limited. Again, this is how we can tune the search to get what you need. In addition to searching for keywords, we can search for patent owners to see what particular patent owners have. We can search on names of inventors. And all of these, again, give us ways of tuning to get what you need. So next slide. By way of example, as Stephen alluded to, there's all kinds of things that we can search for. We can look for, in this example, where they're doing RNP, well, we can search for RNP delivery methods. We can search for the particular enzyme you're using. We can search for modifications to the enzyme you're using. We can search for non-naturally occurring RNA portions of the system. We can search for biolistics. We can search for the regeneration. The key is, do you need that? And I think that that's where, again, we can help you, we can work with you to figure out, and I like to think about this and basket these into that which you must have. That is the most essential and important tier. And honestly, in the this example with the lemon, the CRISPR system is probably not the must-have. The must-have is the trait, is the gene being edited. Because without editing the gene, you have no product, and the product incorporates the gene edit. As in, that lemon is going to be sitting on a shelf somewhere that somebody is going to buy. Your competitor can buy it, a patent owner can buy it, sequence it and say, haha, this says they edited to a gene that I have a patent on editing that gene, and you don't have freedom to operate. So that's really the must-have category of, you don't have a product without it. The next level, which you probably should also search, is what I call the essential to have. It's not part of the product, but it is so essential to getting the product generated, to manufacturing it, to testing it, that without it, you're probably not going to have a commercially viable product. Here again, that might not be the CRISPR system, because there's many different CRISPR systems, and there are the older systems such as Talon, etc. Things that come to mind are the morphogenic promoters, where if you have recalcitrant, hard to transform, you probably don't want to. So for example, if citrus trees, there's many different varieties, most of them are very difficult to transform, you're not going to want to put it into an easy to transform, who cares about citrus variety, and then introgress into trees, that trait for everywhere. You'll want to have a regeneration technology, and if there's a dominant one, that's essential to have a real viable product. The CRISPR systems and the other editing systems really fall into what I call the essential once it's adopted. Those are, again, key to search, because once you've started using it, you have the technology developed. If you do get sued, you can't get a license, you're enjoined, it's going to be a lot of money to get beyond that, and you really probably want to search those. Then the last category are nice to have. This is where a lot of what Steve had mentioned earlier are going to fall into the culture media. Well, there's probably 5 or 6 different culture media. If somebody has a patent, finds out you're using it, sues you, it's not going to undo your whole system. You're just going to swap out one of the other culture media. And so it's nice to have, but you probably don't need to search that. So again, basket into that which is, you have no product, that which you really, really need as essential for getting the product to market. And then those once you've adopted, and this is where, again, I think you have the most options. There's the most input for deciding which one to adopt when you're just figuring out, okay, I have my trait, I want to edit it. What's the editing system? And our recommendation is always talk to the scientists who are going to implement this. Have them come up with a short list of, these are the systems, I know what they are, I know they will work, they're my preferred system, they work better than these others. How much is it going to cost to implement it and how long? Then ask them, what are the other systems that might work? Oh, there's this new one from the MAD 7, it's new, I don't know if it'll work. Probably going to take longer, be more expensive, but at least you have a sense of there's other options if the main ones aren't good. Then in addition, think about what would be the cost of doing with an older system like Talon. Again, getting the time to market and cost of these different options give you a sense of what's the economic value for what you would really want and what you would really need, and which one you would want to say, this is your main thing. Once you've got your short list of, here are the interesting technologies I know will work, here are the ones that might work, then figure out who are the players in the industry that have these different systems or have pieces or components needed for the system. Then think about the freedom to operate. We can do a much more focused and much less expensive, but more information research or information rich if we do keyword searches that might be very broad, but limited to the names of the assignees that have the portfolio to see what do they really have. And then also do maybe a narrow search, but not limited, to see are there any outliers. And that'll give you your short list of, here are the folk to approach to seek and think about a license, think about a valuation license. And that'll, again, do a much more refined and focused freedom to operate search based upon which system you want to adopt. So that is freedom to operate in a hopefully focused nutshell that all made sense. On to licensing.
Thanks, Otis. Let me just introduce myself quickly. Matt Cronin, I'm a partner here in Morrison Foerster's Boston office, and I'm joined by my partner, Wolfgang. Wolfgang, you want to introduce yourself as well?
Yeah, hello, good evening from Berlin. My name is Wolfgang. I am also a partner at MoFo, working out of Berlin and specialized in technology transactions in the life sciences space.
Next slide, Brooke. Wolfgang, I'll lead it. You want to lead it off?
Sure. And thank you, Otis, for these great insights. From a licensing perspective, I think the first aspect to be aware of is that the market is extremely valuable. There are estimates that by 2025, the market will be worth $5 billion. And I think it is fair to assume that a big portion of this will be ag. Otis, do you agree?
Yeah. So we'd sort of bounce this around and thinking about ag is already getting products based upon CRISPR out into the market versus the other obvious uses would be diagnostics and therapeutics. Again, diagnostics are going to be a shorter path, but I think they're not going to be as aggressively pushed out there in terms of getting product to market as we have in ag. And then finally, obviously, therapeutic market. You have to get through clinical trial development, 5 to 10 years. So that's going to be much further out. So I would say, yeah, this is probably predominantly an ag based for that $5 billion estimate.
Okay. And as you have previously alluded, while we are speaking, a tremendous patent thicket is building up in this space. There are estimates that currently over 12,000 CRISPR patent applications are on file, and each month more than 200 new filings are being made. So this is obviously a problem for licensees, as Otis, you have alluded, because in this thicket, you may want to be sure that you only pick what you really need. And another issue here is that competition over these patents is extremely intense, because if you want to be part of this game, of this 5 billion market, you will have to obtain licenses from the patent owners or the IP owners before using their inventions. Next slide, please. But there is good news, and I'm sure that most of you are aware that there are joint licensing frameworks out there that make it easier for license seekers to obtain the licenses they need. Joint licensing frameworks, some people call it patent pools. We don't think that's the right term. It's more a kind of a couple of...
I'm gonna take this guy, I forgot about his 4 p.m.
All right, so joint licensing frameworks. Back in 2017, for instance, the Broad and DuPont created with other players this framework and aggregated basically all that is needed to implement and use Cas9. It has part of the UC Berkeley patents in it too. What it does not include, at least to my knowledge, is Millipore and Toolgen. If you look at CPF1 rather than Cas9, there is also a framework that is available, led by the Broad, aggregating patents from the University of Tokyo, Wageningen, and others. And again, provide for a nice and almost complete bundle if you want to practice CPF1. What is the motivation behind these licensing frameworks? It is obviously to provide access to split patent estates, kind of a one-stop shop. The intention is to accelerate use of the technology in agriculture and drive innovation in this space. And it's also to promote so-called social licenses. Matt will later on speak a little bit about the content of these license agreements, and social license here refers to these clauses in these agreements that you would typically see that are strongly policy driven. And we are also seeing a new trend in the market, and Otis, maybe you want to also chime in here, which is around platforms, mostly startup companies that have developed already platforms that are useful and the basis for collaboration with larger companies to help them fill their pipeline. These deals, however, are then a little bit different than what you would see with the Broad DuPont or the CPF1 frameworks, because it's not just naked licensing. It has very, very strong collaborative elements in it. Otis, do you want to comment on what I've just said?
So yeah, again, these are the newer sort of up-and-comer companies, and honestly, I think that that's one of the side effects of accelerating plant innovation and plant agriculture from these joint licensing frameworks is that in addition to driving adoption of Cas9 and or CPF1 for getting interesting products in the agricultural pipeline to market, it's also driven companies to find new and interesting CRISPR systems so that they can have their own platform. And as you alluded to, again, instead of just saying, hey, we've got this interesting new, maybe it's different from Cas9, different from CPF1, but has these other interesting characteristics that have value. Well, okay, instead of just saying, I'm going to license it separately, it's let's do a collaboration. We will work with you to implement because maybe you're not in the ag biotech space. You're more a traditional breeder, or you've got your own sort of, folks doing molecular biology too, but you want to do a partnership to have somebody who might have more expertise in this new, interesting, different CRISPR system. So yeah, definitely something that's coming along, and it's, again, fantastic to see as as the industry continues evolving.
Yeah, absolutely. And again, I mean, in terms of the structure of the agreements, these work quite differently than the more straightforward licenses that you would get from Broad to DuPont or under the CPF1 estate. And Matt, maybe you want to give us a little overview on what the typical provisions in these more standard licenses are.
Yeah, absolutely. Thanks, Wolfgang. Thanks, Otis. So, I think that the point is we kind of focus now on the actual license agreements that you're likely to encounter in this area, is that a lot of you are very familiar with these license agreements. We know that many of you are not, and so in the limited time that we have, we're going to start at a little bit of a basic level, but really understanding how these frameworks came about and why they came about, I think is really important, given precisely what Otis was talking about earlier with respect to the ownership structure of CRISPR and how diverse that ownership structure is. A lot of our clients that we work with in this space, kind of wonder about some of the terms in these agreements and how they came about, and understanding the underlying joint licensing frameworks. I think it's really helpful in driving the understanding for why these provisions are commonly drafted the way that they're drafted. So Brooke, why don't you flip us over to the next slide. So initially, what we don't have on this slide is what is very common, a free use license for academic research purposes. Wolfgang, do you want to just spend maybe a couple of moments just talking about that before we dive into what folks are likely to see in the license agreements themselves?
Yeah, sure. So many of these institutions offer free licenses for academic research or nonprofit activities. They usually come along under an MTA, material transfer agreement, where these other academic institutions obtain genetic material that has been edited or can be used for that. And these MTAs, again, they are limited for academic or nonprofit research. And a point that we wanted to make here is perhaps some of you have or are operating under these free licenses, but make no mistakes, they are not free in a sense that you can translate those into a commercial context. If you are operating under such a license in your lab, in an academic institution, and think of spinning out a business around that technology that you are working on, you will need to get a commercial license to be able to translate or transpose this research lead into a commercial entity by way of a spin-off or other sort of transaction. This is just a point we wanted to highlight here because the term free license may be a little bit misleading here and can cause problems if you start your company then just on the basis of that arrangement.
Yeah, and so that's a great point, and we can't stress enough the importance of really reading the terms and conditions associated with those, quote unquote, free licenses. A lot of those agreements don't make their way to law firms and to groups like the one that Otis and Mike and Wolfgang and I practice in. They're just entered into without review. And that's not to say that you can negotiate them or edit them extensively, but it is to say you can absolutely understand the terms that are contained in there. And so that there are things that you can do operationally in order to prepare yourselves for when you want to make a move from an internal academic research use only to a commercial license. And those are the things that we hope that folks that are using those licenses are preparing for. And so when you open up the license agreement, you're going to get through the definitions. A lot of definitions perhaps you're familiar with. Some in these license agreements you're unlikely to be familiar with. But then the very next section, typically, these agreements do follow a standard pattern, just like any other license agreement, will be the licenses themselves. And commonly, if you are getting an internal research license, you will see that first. And you'll see some familiar terms that you should really focus on. And again, make sure that the license grant aligns with your intended use of what's being licensed. So this could include the right to conduct research to develop and make products and processes. Certainly that would be abundantly common. Commonly, they come with a fee, so they are fee-bearing, typically non-exclusive in nature. They are commonly field limited to a research field, as well as territory limited, perhaps in some cases. And I think one of the key things to note, and the key things to really read and focus in on, are that a lot of these licenses, many of these licenses, contain extensive restrictions. And those restrictions are critical. And certainly they should be understood, they should be analyzed, and really understood to make sure that the use, the intended use of what's being licensed by you will not infringe upon those restrictions, will not in any way breach those restrictions. And certainly aligning on that is of utmost importance. And then kind of moving on to what the license will contain in the commercial context. It will contain all of those things that you're going to see in the research only license, as well as several additional terms and conditions. And again, here's where we really hone in, and we really want to ensure that the requirements, the restrictions, the obligations, the benefits of the license align precisely with what the company intends to do. So you'll see lengthy provisions with respect to the rights to conduct commercial activities in a particular field, in a particular territory. And every single time we read those sentences, we focus on that language. We want to go back to the terms, how those terms are defined, how the research field is defined, how the territory is defined, how the field is defined. And again, here too, you're going to be subject to extensive restrictions. And again, it's critically important, particularly in the commercial context, to make sure that those restrictions can be complied with and will be complied with so that we don't have any downstream problems. These types of licenses in the commercial context will typically extend to trait users, seed companies, for example, and also subcontractors and affiliates. You will see, as you would in most licenses, you will see a construct whereby there's a whole lot of requirements that you as the licensee will have with respect to permitting use of what's licensed under the license to third parties, such as trait users, subcontractors, affiliates in some cases. And again, really focusing in on what those obligations are. And to the extent that you as the licensee have an issue that you think perhaps there's going to be a compliance problem, really working with the licensor to fine tune that language to make sure that we land at a place where everyone's happy and everybody can agree that we won't have any downstream issues. Unless Wolfgang has anything to add with those thoughts, I will ask Brooke to, Wolfgang, anything to add there? Oh, no. Shaking his head. Okay, I will ask Brooke to move to the next slide. So the next place in a license agreement that you're going to go, usually it's the next provision, which is why we put it next, are the economic considerations. And certainly this is an area where we spend quite a bit of time. And licensors and licensees will spend quite a bit of time focusing on the economic considerations of a particular license. And so initially, there's probably 4 initial buckets of fees that you want to focus on. The license fee itself, which will usually come first. That will typically in these types of license agreements be followed by some sort of a maintenance fee. And keep in mind that the maintenance fee may fluctuate from year to year. The maintenance fee will likely not stay the same over time given that it is commonly based on the number of full-time employees you have that are utilizing the licensed technology for the purposes of the license. And so that's just something to keep in mind, particularly from a budgeting perspective. As we think about what is the total economic impact of these license agreements to the licensee, we just want to keep in mind that there is fluctuation in some of the fees that in maybe a different type of license agreement would not fluctuate. So that might be a little bit of something that you're not familiar with. And then when you move to the commercial context, certainly you're going to be talking about milestone payments and really focusing as the licensee on what those milestones are, the timing for those milestones, the timing for those payments. And then of course, we wouldn't be doing our job in the context of a CRISPR license if we didn't talk about royalties. And in the commercial context, you'll spend quite a bit of time talking about royalties and the amount of royalties and the payments of royalties, which brings us to the next two topics. So when you think about kind of the total economic impact, there's some other provisions of these license agreements that do impact the licensee and the licensor economically. One topic of a lot of conversation these days, and certainly you can Google this and find quite a bit of information on this, is the concept of reach-through rights. And we do talk a lot about this. And one of the things that both the licensor and the licensee will want to spend a lot of time on is working through the applicable definitions, like, for example, the definition of traits, to get comfortable with the application of royalty payments throughout the entire life cycle of the license. And one of the things that certainly is talked about quite a bit is the ability of the licensor to collect royalty payments on not only when the licensee identifies a trait using the licensed technology, but also in the event that the licensee were to create a product that incorporated that trait, but was created without using the licensed technology, perhaps using some other gene editing technology, or perhaps possibly plants that are made or bred through conventional methods. And so we talk quite a bit about the application of royalties holistically. And there's lots of terms like the definition of traits, like the definition of royalty, like the definition of net sales, where you really want to focus and get comfortable with the application of royalty payments throughout the life cycle of the license. The second area of really particular interest when it comes to thinking holistically about the economic impact to the licensee of these license agreements is the term of the license. And we talk about the royalty term as another issue when it comes to reach-through rights. And so on that, I will turn it over to Wolfgang to talk a little bit about what the conversations we typically have, the negotiations we typically engage in between licensors and licensees, and perhaps where we're comfortable landing on this particular issue. Wolfgang, back to you.
Yeah, thank you, Matt. Maybe we go right to the next slide, which has kind of a illustration of a situation that you can potentially encounter when assessing the royalty term in your CRISPR license. Usually, the royalty term provision says the later of patent expiry or some period to begin negotiated after first commercial sale. But as Matt has mentioned, the definitions of what is a licensed product are critical here, because it may happen that you encounter a situation where the creation of an edited gene using the licensed technology, which later on goes into the licensed commercial product, has been created a few days before the expiry of the patent. Then you only start usually a rather long development phase going through proof of concept, and then ultimately first commercial sale, hopefully, if everything goes well. And some of the provisions we have seen would then result in a royalty payment kicking in long after patent expiry, triggered by the first commercial sale and running for a to be agreed upon time after that moment. It is a typical structure if you talk about research tools, because these patents do not necessarily read on the final product. But what you want to make sure is first be aware of how this can play out and what the impact on your business case is. And also discuss with licensor whether there are ways to pick up some of the loose ends here and maybe define maximum terms or something like that, which we have also seen in other agreements. For instance, commercial sale must have happened within a certain period of time after patent expiry. That would be one of the solutions here to get more certainty or clarity around what exactly the commercial impact of the license provisions and the royalty term on your business case would be. Matt, back to you. We have 3 minutes left, or Mike, do you, we have another slide that we could cover.
Yeah, why don't we do the final slide and then we can talk about the questions tomorrow.
Sure. So that's great. Finally, you know, we dropped in some additional notes here on the final slide, and I hope everyone understands 25 minutes to cover CRISPR licensing is not a lot of time, but I hope, Mike, we can schedule a deeper dive on all of the different licensing issues in the future with this group. We'd love to do that. But a lot of the literature that you'll read when it comes to licensing CRISPR and Cas9 and CRISPR technologies is that licensors, and we agree with this, have really done a lot of work to, you know, structure these agreements as not to be cost prohibitive. The idea being, and we could go way back to one of the earlier slides, particularly with respect to what Otis was talking about. The idea being is that they want the technology to be used and developed, and for new technologies to be developed. There's a lot of social impact and social awareness that comes from this technology, and so making the economic terms non-cost prohibitive, I believe, and I think the literature supports historically, was done so that it is not cost prohibitive. And certainly a lot of work can be done between licensors and licensees within the economic framework of these license agreements to provide for that. I think that licensors have really implemented what they perceive as fair. And we can absolutely talk about that. I would love to talk more with many of the folks on this group about in this group about that particular point and whether that's their perception as well. But certainly I think that from a licensor's perspective, the economic structure and the licensing structure is what they perceive and what they've landed at as a fair approach for licensees. You know, noting, of course, that in many cases, not many cases, but in several cases, royalties are not payable in certain circumstances. And you know, you can certainly read the sections of those agreements that apply, particularly with respect to products to smallholder farms, which is defined in these agreements, particularly with respect to products that are sold in developing countries. And there's a lot of social impact awareness that's very evident in these license agreements. And that's from our perspective, a good thing for both licensors, licensees, and the CRISPR community in general. You know, that being said, there are other economic terms, and I know we're at the top of the hour, so I'll drop this bomb and then we'll probably have to pick it up tomorrow. But there are other economic terms in the agreement that you really should pay attention to, particularly with respect to whether or not royalty stacking applies, those circumstances where there are reductions of royalties, circumstances around the impact on royalties to patent challenges, termination rights for some non-standard things that you as licensees might not be familiar with, what's defined oftentimes as material negative impact or environmental impacts. So there are other terms that are kind of bundled in the economic construct of these contracts that are really critical to pay attention to as licensees, and certainly to the extent that there's a concern engaging with licensors on these terms. So I'm so sorry we have to leave it at that, but Mike, I'll turn it back to you to wrap things up.
That's great. Thank you, Wayne, Steve, Otis, Wolfgang, and Matt for a great session today. We'll see everyone back here same time tomorrow, and we'll continue the discussion. Thanks very much. Bye-bye.
Thank you.